Hello, my name is Preston Arnold, and my partner is name is Dalton, and we decided to do sliding filament theory for our final project for the exercise physiology lab. And so he did the illustrations and the typing on GoodNote, and I'm doing the voiceover for the project. So to start it all off, I have the general idea of the muscles and all the components that go along with it to help better understand what's happening in this process. So to start it all off, here's the bicep muscle right here. And then if you open up that view a little bit more, you can take a look at here's the whole muscle. And then each of these little red circles are what we call fascicles. So then if you take the fascicle and open it up and expand it even more, you have each little individual muscle fibers. Then you take the muscle fiber and you expand it out even more. You have the muscle fiber with the acetylcholine receptors. And then if you take this muscle fiber and expand out even more, you can really get a good look at the myofilaments. And this structure is called the myofibril. So sliding filament theory cannot be done without the use of calcium. And because this project is only about sliding filament theory, I thought I'd just give a general idea of what's going on with the neuromuscular junction, which would explain how we get calcium to even go on with the process of sliding filament theory. So first off, we have the spinal cord and we have the brain. The brain sends action potential. The action potential travels down the motor neuron and releases acetylcholine, this is basically our motor neuron right here. This acetylcholine binds to ACH receptors. This allows for sodium to move into the sarcolemma and it allows it to depolarize, but it needs to go deeper. So it goes deeper into the muscle through T-tubules, and then once it depolarizes deep into the muscle, it tells the sarcoplasmic reticulum to release calcium. And sliding filament theory cannot be done without calcium, so that's why I gave a little summary of what's going on in the neuromuscular junction. So down here, we have the sarcomere, and the sarcomere is the smallest functional unit of the muscle. So right here, we got the Z-disc on both sides. Z-disc they are binded with titin, and titin keeps myosin attached to the Z-disc. And so right here, we got myosin with the myosin heads, and we have actin, and this little blue winding structure is actually tropomyosin. Okay, so down here now, we have the tropomyosin complex, then we have troponin. And troponin acts as like the key lock that, until it's on there, until the complex is gone, the myosin head cannot bind. But with the presence of the calcium, that's the whole point of the neuromuscular junction, it binds to troponin. And once it binds to troponin, the tropomyosin complex releases and it actually opens up this binding site right here, which allows for the myosin heads to bind. But the myosin head cannot bind without the presence of ATP. So what happens is ATP binds to the myosin head, and just for reference, because there's no labels, this is the myosin and myosin head, and this is the actin. So ATP binds to the myosin head, then ATP dissociates and forms ADP and phosphate, which is right here. Then this allows for the myosin head to cock back in position and bind to actin, which is the third step of sliding filament theory. So then next, ADP and phosphate are released, and this causes the myosin head to go back into position, as shown in this small illustration. This is known as the power stroke. So then next, the ATP molecule binds back to the myosin head. This releases the myosin head from actin, and then the whole cycle repeats all over again. So they go back. We got neuromuscular junction, releases calcium. Calcium binds to troponin. Tropomyosin complex releases, opens up active site. ATP binds to the myosin head. ATP dissociates and forms ADP and phosphate. ADP and phosphate bind. Then they release, allows the myosin head to go back in position. That's the power stroke. And then ATP molecule binds the myosin head, and this releases the myosin head from actin. and then, like I said, cycle repeats. So that is sliding filament theory, and I hope the class or the viewers enjoyed. Thank you.